We had 19 dolphins that had wounds worth documenting. Some were from shark bites, like the one you see here on a speckled female named Podrick. Others were deep gashes like we saw on Bijou, which was above her left pectoral fin. We also saw that Nugget, still in the two-tone H class, had lost his right eye. Because of this wound, he either turned inverted so that his left eye was facing you, or would just turn around completely so you were on his left side. Some of the other wounds we saw could have been inflicted by other dolphins during aggression, but one injury in particular had us wondering what happened, and it's possible it could have been an orca attack. Depending on where orcas live, some of them prey on marine mammals, including cetaceans. According to a paper by Dunn and Claridge in 2013, the orcas who live around the Bahamas have been sighted preying on Atlantic spotted dolphins. And historically, the orcas have been sighted in the waters near Bimini. The wound we believe could be from an orca attack was on a bottlenose dolphin and not an Atlantic spotted dolphin. But the spacing between the teeth marks are too large to be from another bottlenose dolphin. If this wound is not from an orca, another possibility could be a false killer whale, since these animals are also in Bahamian waters. Hopefully next summer we won't observe so many wounds on the dolphins we study. Thanks for tuning in for our 2021 field season update. If you would like to experience a trip out on the Stenella, please visit our website and click on the Participate tab. Then click the Bahamas trips and all the necessary information is provided there for you. We hope to see you out there with us next season.
Hello, everybody. I think we're going now. Welcome to Wild Ocean Science. This is the fourth time we've done this, the second time virtually. And it's a great pleasure to have all the Wild Dolphin Project team back in the game here of educating us all about these wonderful, interesting, endlessly interesting animals. And so glad they had a season and a chance to get on the Stenella and see the animals. Lots of questions raised by, gosh, awful lot of dolphin activity going on there with all those pregnancies, but also the injuries and the orcas. A lot of questions there. I didn't even know there were orcas in the Bahamas. So we're going to learn more about all of that. Everything you're going to hear today comes from the hard and good work which began with Denise Herzing 32 years ago. She started at the age of 2. She was very precocious child. And in any case, this is work that is vital. It is unique. Nobody does this, and it's not cheap. So part of this is a two-way street event. We want you to learn some things, take some stuff on board, learn what's going on out there in the Grand Bahama Bank. But we'd love for you to contribute as you go along. And there's a lot of ways to do that. On the webpage itself, you can just click the donate button. You'll notice there's a bar at the top. We're right now at $1,130. I chipped in right before we started here to prime the pump, but we'd like to get to 10,000. So let's try to move that bar to the right. You can also text the word biology to 243-725, 243-725. Or you can just give the office a call. There's operators are standing by, as they say, 561-575-5660. So chip in and help out. And the more you learn, the more you should participate at any level you can, including contributing. So Denise, so good to see you. And I'm so glad you got underway this year. I know, it was kind of touch and go. Tell us a little bit about, were there particular COVID challenges you had to address, or was it fairly straightforward?
Well, as probably everybody knows, we missed most of our season last summer. Well, the Bahamas, it was open. We had to do testing, as many countries do, but we managed fairly well. And, we had some field time, a lot more than last year, which was great. But, what's great today is that we're going to hear a lot of the details about the analysis of the data. because a lot of people know us from the field work and from beautiful images and films that have been done telling this story. But what we do in the off season, we don't just sit around and wait for the next field season. We're analyzing data that includes students and staff, and that gets to the real meat of what is going on with these dolphins. So it's not just pretty images, it's about how healthy is their population, how do they reproduce, where are they moving, are they finding enough food, all those questions. And you're going to hear from 4 fantastic young researchers who've been working with us for many, many, many years. And I hope everybody will learn a lot. So I'm excited.
I am certain that is the case. But yes, we should dispel the notion that you sit by the pool with drinks with umbrellas in them all in the off season. This is when actually, in many respects, the real work happens. The papers are published. The data is compiled. None of it means anything if you don't turn it into science. And that's what this is all about. And, you know, this group today, it's like an all-star reunion for the Wild Dolphin Project. Some of your greatest research assistants, interns, scientists who you have touched and mentored along the way, Denise. It's got to be a thrill seeing them move out into the world and do their own projects and flourish, and then to have them come back and share what they've learned along the way is it must be kind of fun.
Yeah, it's great. And we try to keep a few of them really close too. We don't want to lose them, but you know, they have lives elsewhere and exciting projects on their own. So yeah, we're going to hear a lot about how they're still interacting with the project and working with it. Yeah, looking forward to it.
So each of them has put together excellent video summaries of some of their work and where they are. And we're going to begin with Cindy Elizer, who was a big part of the operation for many years. She was a research assistant from 2002 to 2010. She's now on the West Coast looking at what's going on in the Pacific Northwest. As it relates to sea mammals there. Let's take a look at her video, and then we'll bring her in. We'll have a discussion with her about what she has learned and is learning. So go ahead, Melissa, and roll the video.
Spotted dolphins have a complex society that resembles that of bottlenose dolphins, chimpanzees, and even humans. They live in groups that constantly change, but have long-term relationships that are usually correlated with age, sex, reproductive status, and kinship. For example, males form alliances, which are like best friends, often for life, and female relationships revolve around reproductive status. Calves have to learn the rules of the society to figure out what they can and cannot do, and should not do. Sounds familiar, right? These social dynamics are integral to the health of their community and how they are able to adapt to changing conditions. So what happens when individuals move away, move in, or die? When at the individual level, that is a normal part of community life. But what if this happens on a large scale? In 2004, 2 major hurricanes hit the Little Bahama Bank, or LBB study area, within 3 weeks of each other. These dolphins had never lived through an environmental disturbance like that before. The result? 36% of the population was lost. This caused some minor shifts in the social structure of the remaining dolphins, but interestingly, their core community structure remained: the socially, genetically, and geographically distinct northern, central, and southern clusters. And they remained on Little Bahama Bank, their home, for decades. Thus, it was a huge surprise in 2013 when the entire central cluster and a few individuals from the northern and southern clusters relocated 161 kilometers south to Great Bahama Bank, or GBB, which was already home to resident communities of spotted dolphins and bottlenose dolphins. How do 52 dolphins move into an already stable community on Great Bahama Bank? Do the 2 communities merge into one or remain separate? Social structure can influence behavior, movement patterns, and ecology, which in turn can affect the survivability of the individuals and their community. How these individuals can live and survive together on Grand Bahama Bank lies in the social associations they forge. For the first 3 years after arriving, there was little integration between the LBB and GBB communities. Imagine a huge group of what you thought might be tourists, just ended up staying and making themselves at home, with no intention of leaving. It would be a little jarring to say the least. This network diagram shows social connections between individuals, and as you can see, the 2 are quite distinct, with just a few connections linking the 2. Are those dolphins forging a path for the two communities to come together? Only time would tell. Over the next 3 years, connections increased and associations changed, but it is not as clear-cut as everybody joins up into one big happy cluster. As you can see in these network diagrams, this community, now made up of LBB and GBB individuals, has divided into 3 social clusters, where the new relationships are influenced by their previous associations and the choices of the individuals. Some GBB individuals have remained more separated in the cluster with only GBB dolphins. Others have forged connections with LBB individuals, forming 2 mixed clusters, one with mostly GBB dolphins and one with mostly LBB dolphins. This structure continues over the next 3 years, and you can see the increased connectivity between the clusters. Although these connections have increased and strengthened, their associations are still stronger within their original communities than between. For example, LBB dolphins have stronger associations with other LBB dolphins than with GBB dolphins. There is definitely a link between the GBB only and mixed mostly GBB cluster, as you can see in the diagrams, with movement of GBB dolphins between those clusters fairly common. In contrast, LBB dolphins only moved between the mixed groups. Male alliances were present, some long-term relationships from after the hurricanes, and others new, even one between 2 GBB and 1 LBB individual. How the integration is occurring is being shaped by these new relationships formed after the move, along with those long-term and familiar relationships from their previous communities that they have forged over many, many years. Assimilation takes time, and while new relationships are being made, old ones remain that may continue to influence the social restructuring of this new community. This social flexibility is important for their long-term survival as environments continue to change and upheaval events occur. What the future holds for this new community and what the social structure will look like depends on how these social relationships continue to develop as these dolphins live and thrive together on Great Bahama Bank.
Fascinating video, Cindy.
Thanks.
Cindy Elizer, who normally is in the Pacific Northwest, happens to be in New Mexico. That's a long story. It doesn't matter. But there are so many questions that come out of this, but I know that Denise wants to start because she's so happy to see you. So I got to ask one question. How on earth did you just do that? I mean, the amount of time it took to identify those relationships, it's a little daunting, isn't it?
Yeah, well, I mean, that's the power of Denise's study we talk all the time about photo ID, and it's so nice to know the individuals, but getting this information you cannot do without knowing each and every individual and who they're related to and who they hang out with. And so all that work that goes into getting out there and taking those photos then goes back and you ID them all, which takes its own amount of time. And then you pulled into the data, you put in the Excel sheet, and you have these programs, and it takes a few months, if not longer, to really sift through everything and put it in a diagram that makes sense, right? So a lot of it is it's looking at all these relationships, and it's very complex. So how do you make it in a way that you can show it to somebody and go, oh, that makes sense? Because sometimes it's very hard.
So just for everybody's background, so Cindy did her master's thesis and her dissertation on the relationships of the spotted dolphins and some bottlenose on Little Bahama Bank. For decades we were there. That was what her work was. And when we had this major displacement down to Bimini, we called her in and said, oh, would you like to analyze this new thing that's going on? So my question to Cindy is, did you ever imagine you would be analyzing the associations of 2 different societies? I mean, I don't think I envisioned it.
No, I mean, and I always, as you talked about before, wanting to keep close, and I always want to be as involved as I can with the project because I miss it and I miss you guys. And so this is a way to be able to do that. But I was envisioning, what's happening on Little Bahama Bank and, within that realm of how those relationships are shifting or changing or whatnot. And we saw some changes after the hurricanes, but it wasn't major. So when I heard that they moved, I was just like mind blown. Like, well, they didn't move after the hurricanes, which is where you would think that might happen, where things got so disrupted, but they didn't. So it's like, okay, these guys are going to be here. They're not going anywhere. They've been there for decades. And so to all of a sudden be like, they're gone and they're there now. And it really brought up in my mind, like, oh my gosh, the social structure part, what what happens when you have an entire community move into another community? Like, will that even work?
And
I just love. I've got to. Let's get a question in here, Denise. What I love about this is the serendipity of it all. Right, the fact that this thing happened, and you've taken great advantage to learn a lot of things you never could have if it didn't happen. And there's so many questions that come out of this. That's what makes science so interesting. Gretchen is asking this for you, Cindy. Are there any differences in assimilation between males and females?
So that's a good question. I haven't been able to get through that level of analysis yet. But in the after the hurricanes, there was some differences between males and females in who they allowed into the community, and there is differences in other places where you have males and females have a different ability or want to allow new individuals to come in. So there could be some level of that.
So Gretchen, you might want to offer up a little donation. Maybe that would allow Cindy a little bit of time to go look through her data a little more. Denise,
Right.
Go ahead.
well, I was just going to point out the reason we think the dolphins moved from Little Bahama Bank down south was because of the food chain. We saw some serious oceanographic features that fell off the scale, basically a drop in the food chain. And so the reason they moved, we believe, is lack of food in their normal area. And that's also increased pressure, right? So it's not they're just moving for social partying or something. They're hungry. And so now they're competing with the resident group potentially. How do you adjust that spatial thing and who gets the choice feeding grounds and who doesn't? So it's fascinating stuff. And it's a natural experiment. Sad for them, but we're just the storytellers, right? So it's pretty fascinating.
And that's what happened with the hurricanes too, with my dissertation. Like the hurricanes came through and it was just this experiment that we could do when you wouldn't be able to otherwise, to see what would happen after you lose that many animals. So it's great that we're out there that long, Denise is out there that long, that we can use these unique situations that happen to learn so much more than we could otherwise.
You got to show up and you got to be ready to go with the flow, as it were. So, and with the immigration. So we're going to continue on this subject of the immigration and issues relating to their range, which is also a fascinating tale. We will go to our next Wild Dolphin Project All-Star on this great alumni show. Brittany Hill was with the project. I think she started in 2018. And she's, she drank the Kool-Aid and is not coming back, as it were. So let's look at Brittany's video, and then we'll bring her into the mix.
Hello, everyone. My name is Brittany. Welcome to my presentation, Immigrant Dolphins, a shifting home range and Atlantic spotted dolphins in the Bahamas. Home ranges are defined as the area utilized by animals for gathering food, mating, and caring for young. So why do animals maintain home ranges, and why do we study them? They do so because site fidelity, or the tendency to return to a previously occupied location, provides a fitness benefit. Understanding animal home ranges reveals many features of a species' ecology and behavior. Animals do not use their home ranges evenly, as resources are usually heterogeneously distributed. Animal distribution depends on maximizing access to resources and minimizing competitive interactions. If an animal cannot meet its needs living on resources within its home range, it will move and go to another location. So, what do animals encounter when they move to a new home range? Immigrant animals may encounter a difference in access to resources and habitat quality. Inter- and intra-species interactions may occur, including competition, which could decrease access to prey, and increased aggressive encounters, as you see here. While home range shift studies are more common for terrestrial species, they are lacking for dolphins and whales due to the difficulties of tracking and monitoring these populations. As you just heard from Cindy, in 2013, the WDP documented an unprecedented mass immigration event in the community of dolphins monitored for decades on Little Bahama Bank. When immigrant LBB dolphins moved to GBB, they not only acquired a new habitat, but also encountered resident communities of dolphins. My research aimed to determine if there were significant differences in immigrant and resident home ranges, and if competitive interactions were present to drive changes to immigrant home ranges. The overall question was, how did the emigration event affect the home range of the immigrant dolphins? I used geographical information systems to create home range models to answer this question. I first compared the home ranges and core areas, which are areas of increased use within the home range, of the immigrant LBB dolphins who moved to GBB, shown in orange, to the resident GBB dolphins, shown in gray. There were 16 immigrant dolphins and 23 resident dolphins, sighted over 20 times on GBB from 2013 to 2020. The graphs show that immigrant dolphins maintained 43% larger home ranges and 42% larger core areas on average when compared to resident dolphins. Next, I conducted a paired comparison of 13 individual dolphins' home ranges and core areas when they were residents on LBB to when they were immigrants on GBB. Paired tests showed that the home ranges and core areas were larger for these dolphins as immigrants than residents by 34% and 94% respectively. For example, Tyler was an adult female living on LBB who moved to GBB in 2013. Her home range increased from about 130 to 230 square kilometers, and her core area increased from 17 to 47 square kilometers. Overall, you can see that immigrant home ranges on GBB, shown in gray, are larger and have more fragmentation than resident home ranges, outlined in black. The immigrants' range is also shifted to the north, likely for access to the deep water edge where they nocturnally forage. Next, I compared the behavioral frequencies for resident, immigrant, and mixed groups of dolphins. On GBB, the immigrants were more often found in mixed groups where they interact with residents, while residents were most often found in resident-only groups. This graph has a lot of information, but the major takeaways have to do with aggression, foraging, and courtship. Aggression occurred more frequently in mixed groups of resident and immigrant dolphins when compared to resident-only or immigrant-only groups. And while aggression increased, foraging decreased. These appear to be costs associated with the emigration event for the immigrant dolphins, who spend the majority of their time on GBB in these mixed groups. Courtship occurred more frequently in mixed groups than immigrant-only groups, a benefit of immigrant integration with residents. Additional analysis showed that immigrants on GBB are inhabiting deeper waters than residents and have less access to their preferred bottom type, indicating that immigrants are using lower quality habitat. In conclusion, immigrant dolphins maintained larger home ranges than resident dolphins and lower quality habitat. Immigrants face integration challenges with residents, including increased aggression and decreased foraging opportunities. It is likely that competitive interaction with residents drove immigrants' larger home range requirements. Although species home ranges are not static, fluctuating over time to remain within favorable environmental conditions, Earth species are being redistributed at accelerating rates due to climate change and other anthropogenic causes. Studies of this nature are critical for cetaceans, as they are particularly vulnerable to the impacts of climate-related redistribution. Emigration is a risky behavior that is still poorly understood due to the challenging nature of long-term studies. Range shifts caused by a rapidly changing environment will be more complex than predictions based on climate variables alone. Non-climatic factors such as the strong effects of local competition in new areas should be considered in management and conservation decisions.
Hey, Brittany.
Hello, everyone.
So, nice work, as you know. So important to document those details. I have a question. You just went through a major displacement yourself, moving to Hawaii. So do you have any things you'd love to analyze from your perspective as a human, changing your home range that you wish you could get to more detail? I'm curious.
Human home range shifts. I think I'm pretty used to it by now. My husband's in the military, so I guess I would be curious for how the home range shifts would differ for people who are used to moving versus someone who's moving for the first time. So I'm sort of in that transient population versus a resident population.
That's funny. That's funny. Yeah. So you have getting to know new neighbors and competing for grocery store food and that sort of thing.
Yeah, there's a lot of people on a small island here. So, yep.
Hey, I'm back. I'm sorry. I don't know what happened, but wow. I got to say, now I know, Denise, you were co-advisor on that thesis, right? But just so everybody knows, that's a master's thesis. I mean, that's an impressive piece of work that you put together there. I assume you got an A, right?
Yeah, A plus. Probably.
Absolutely. Absolutely. And again, the amount of, we should back up and remind people that the first name of this organization is WILD. These studies, you can't do these in a tank somewhere, right? There's some things you can learn about dolphins in captivity. God forbid, we don't love that idea. But there's no way you would know about migrations and immigration patterns and ranges. You have to get out there on the ocean and do this hard work, which is not easy or inexpensive. So think about that as you understand these insights, which you couldn't get any other way. Brittany, again, I got to ask you a little bit about the process there. How hard was it to put all this together? It seems like an awful lot of data crunching.
Yes, there was a lot of data crunching, a lot of work in Excel and several other programs like GIS and the geospatial modeling environment. But again, that's the power of computers and technology today. We can input all of this information and run these models. There's certainly a learning curve there. It was new for me, but it's really incredible what you can learn from this data. And as Cindy mentioned, the power of Denise's study, that we actually have this long-term data and not just photo ID from the surface, but we know the animals' relationships and their behavior in the water. So we can not only just map a home range from sighting locations at the surface, we can do a home range and then look at what is the bottom type, what is the depth, which immigrants or residents are getting to use more preferred habitat and things like that.
We have a question from Bill. Did depth preference change for the immigrants versus the residents?
The depth was different for the residents versus the immigrants. So we found that on GBB, the residents were inhabiting shallower waters than the immigrants. And this provides more protection from predation from tiger sharks and bull sharks and things like that. So more protection and easier detection for the dolphins in shallower waters.
Denise, I missed part of it. So did you have any more you wanted to talk with Brittany before we move on?
No, I was just going to point out that the shallow water, it's kind of like the grocery store with potato chips for the dolphins too. Like they have all these like yummy fish under the sand, and at night they go off this deep water edge. So it may be that they're getting access to more snacks during the day, the resident guys, and that's important for metabolism, caloric values, that sort of thing. So I'd hate to be without potato chips myself.
One more question for you from Liam. Did home ranges differ between males and females of the different groups?
I started to look into that. I was a little bit beyond the scope of my study, but an initial look at that, I didn't see any major differences between males and females.
Okay, there's another little piece of research that we can fund, people. So let's get busy here. I want to thank, by the way, Rita Golke and Rebecca Smith and Jody Volker, who all have contributed while we've been talking here. But let's move that bar up at the top here a little bit as we continue our discussion with the WDP All-Stars. And that's not like a wrestling match. That's actually, we're talking science here. So let's move on to our next All-Star. Michelle Green, who is currently at the University of South Florida in Tampa. And she was at Florida Atlantic University in 2008 and spent quite a bit of time with the Wild Dolphin Project as a graduate student and a research assistant. Let's look at her video and then we'll bring her into the discussion.
Have you ever looked at a group of animals and wondered whether the animals were related? Have you ever wondered if you were looking at siblings, parents, or aunts, uncles, and cousins? Or maybe they aren't related at all. I often look at the dolphins and wonder about those relationships. I'm Michelle Green, and I've been heading up the genetic research for the Wild Dolphin Project for several years. The genetic work is really exciting because it opens up a whole new understanding of the wild dolphins. Because dolphins are so social and long-lived, we can use underwater observation to tell us a lot about the animals. In particular, we can determine maternal relationships through extensive observation. Mothers and calves are always observed close together and engaging in behaviors like nursing. Because the research team spends so much time in the water each year, they can observe and track new baby dolphins. Over the decades, this provided a fairly complete maternal family tree. We know mothers, calves, siblings from the same mother, and so on. But we could never pin down the paternal side of the family tree. And that's because dolphins use courtship and mating behaviors for purposes other than reproduction. Mating and courtship behaviors can be seen in many different types of encounters. Given the fact that so many pairs of animals engage in courtship, it's truly impossible to determine which male is the father of a particular calf without doing a genetic study. Knowing the genetic relationships among the animals is important for many reasons. It allows us to contextualize the behaviors seen among a group of dolphins, and it helps us understand just how much genetic diversity we have among our population, and that's an important part of their longevity and resilience in the face of a changing environment. But paternity is an especially interesting facet because we can determine whether a few males are siring the majority of the calves in the population, or whether many different males have the opportunity to sire a calf or two. As you know, the Wild Dolphin Project doesn't touch the animals, so you may wonder how we get DNA without touching them. The answer is in the poop. It's a non-invasive, opportunistic method whereby we can collect fecal material and isolate the dolphin's DNA. And undoubtedly, your next question is, how do you collect the feces? Every time researchers enter the water, they carry small cups in a neoprene weight belt. In the event that an identifiable animal defecates during the encounter and the researcher is close enough, they stop and scoop a bit of the poop into the cup. It's quite a feat. The cups are brought back to the boat and allowed to settle out before they're transferred to a stabilizing buffer. At the end of the trip, the samples are moved to a genetics laboratory for storage and processing. In order to look at paternity, we need to collect samples from the mom, the calf, and from all of the possible dads out there, which is a lot. Given our past work, we found that older males seem to sire the calves, not the younger males. Since then, we've collected many more samples and the population has undergone some major changes. Now we're working on the new samples to see if the pattern of older males siring calves holds true. And we truly appreciate the support as we continue this important part of the research.
Totally fascinating. I guess people out there, when they see this, probably should give a shit about science, right? I mean, really, it's right.
Oh, Miles.
They do. We hope
Sorry,
they do.
Low-hanging feces there. I had to go for it. Good. Good to see you, Michelle. And really, that is, it's so interesting. This whole idea, I'm kind of fascinated by the process that you guys really don't miss a trick, do you? And I mean, it's not like there's not like a dolphin genome project that I'm aware of. Is there? Has anybody ever done the full genetic sequence of dolphins?
No. And when we were first starting this work, the big question was about relatedness, but even then people weren't convinced that there was a way to get DNA from animals without having to take a biopsy from them. And so the big hurdle that they were going through at first was, well, what tissue can we get our hands on? It happens to be fecal material. And is that a reliable source of good DNA for these types of studies? And it turns out it is. And then there's
one quick question, because I we have who's joining you, Denise? Is this another research assistant?
Zorro, yeah, he's my research assistant at home.
Anyway, So quick question though. I'm sure this is a really stupid question, but sometimes, that's what I do. The stupid questions. How do you know when you're examining DNA found in feces that it is not some DNA from whatever that particular animal ate? Is it quite obvious that the DNA is dolphin DNA?
Yeah, that's a great question. And that's one of the challenges of working with fecal material is you can pick up the DNA from what they've been eating. So we're targeting the sloughed intestinal cells that get sort of scratched off as the feces passes through their body. And then we can use what are called primers in our reactions that are specific just to the dolphin DNA. So we design them to match the dolphin DNA and not the fish DNA, so we can selectively pick out the dolphin signature.
And presumably there's being a mammal versus a fish, there's pretty dramatic differences, I imagine, right?
Yes. Yeah. We actually, with good primer design, you can easily tell the difference.
All right. Denise, you want to chime in?
These are designer genes. Oh.
That's right.
We have a question here from Nancy. In other sentient species, is it common for older males to sire the majority of the young? If dolphins have sex for other reasons, as well as reproduction, how is it that the older males are often the sire? Is it frequency?
Yeah, so we should clarify too what we consider an older male in this scenario. So we're
careful about that. As an older male, I'm a little nervous about where you're going with this, but go ahead.
So you guys may know of the different age classes that the animals go through. So the older males, we consider those males the ones that are fused and older. So they're usually fused around age 16 and plus. And then the mottled males, they should be capable of siring males, but we haven't found any yet that have sired a calf. We're working on new samples now because, of course, like we just said in the first video, there's 18 new babies on the horizon for this coming year. So every time we have new young, there's an opportunity to assign paternity.
Exciting times. Denise, go ahead.
Yeah, so Michelle, obviously paternity is the big focus because that's the only way to get this information. But genetics is kind of one of those black boxes to a lot of people, kind of like machine learning. It's like, ooh, the magic box, what can it do? So there's a lot of new cool stuff people are doing with genetic analysis. Do you have any favorite ones you're eyeing for our future here?
Oh yeah, absolutely. So the genetics, it's really interesting. As I watch through all of the different studies that my colleagues are doing, the genetics can plug into that as a new sort of facet of understanding what they're already doing. So just knowing the relationships among them is important. But with the new work, I think that the most interesting thing that's come up, right, with this big shift of the animals moving down south is genetically then what happens to the diversity of our populations. We know climate is changing, so there are cool next gen tools that people are trying really hard to get to work with fecal material that I would love to, be on the forefront of that. And the next gen work, as Miles mentioned, that allows us to get into that whole genome sequencing type of work where you can start exploring more about actual genes, which genes are expressed, do they get turned on and turned off in certain animals and in certain situations.
We have a question from Charlie. Charlie wants to know, is there one male who has sired the most offspring, the super dolphin stud? Who is that?
We don't have one yet. We don't have one yet. So it was interesting though, because that's a great question. These older males, there were a handful of them, not even a handful, 3 of them where they were assigned more than one paternity. So we're at a tie right now, where a couple of them we know of 2 offspring that they definitely sired based on the genetics. And so there's a few. One of them has the name that is fitting. His name is Romeo. So we always throw him out there.
Of course.
As a living up his name.
But wouldn't you say, Michelle, that, Romeo and Big Gash, they were coalition, right? So that's coalitions may have some rank as far as producing sires, right?
Absolutely. And so with Cindy's work on associations and knowing which males are in these sort of wingman types of relationships and they're going after females, It's interesting that once they might have that sort of relationship solidified, it might make it so that they are more competitive when a female is actually receptive and ready to have a calf again, right? Because if a female already has a calf, she's raising a calf, then right now she's busy with that young one. It's going to be a couple of years before she's ready to have another one. So again, there's just these small windows of time where the males would be using mating and courtship for actually having offspring, and that alliance partnership could definitely be a contributing factor.
Would you describe dolphins as promiscuous?
Yes, I would. Yes, but not in the way that we think of promiscuity, right? Because it's, the mating and courtship behaviors are just a part of everyday life. I explain it to people as in, in a similar way that you and I, before COVID, we might introduce ourselves and shake hands. It's a very similar kind of vibe for dolphins with mating and courtship behaviors. It is really just part of everyday life. They teach it to the little ones, the calves at a very early age, because it is so ingrained in the types of behaviors that they do. But again, it makes my job a little bit harder because there's absolutely no behavioral basis that can go along with being able to assign paternity. We know that there are lots and lots of courtship behaviors going on.
It sounds like it's really cool to be a dolphin. Let me say that. All right. So Haley wants to know this. Is it possible that the older males have a genetic component that makes them more likely to sire offspring, or is it just experience and or intimidation of the younger ones?
That is a good question, and one that would require something along the lines of that whole genome sequence and gene expression work to see if it's really truly a genetic component. There's nothing that we can see on the surface about like how they look, like they're extra spotted or have some type of, physical characteristic that the females might prefer. So genetically, yeah, we would need to sort of dive into the genome on that. From this point of view, what we know right now, since we can't get at that genetic question, it's very likely that it is some sort of experience level. That Denise could potentially talk more about. Just the older males, once they're in an alliance, they just act differently than the younger males who are still, fighting and aggressive and figuring out who their alliance partner is going to be.
Yeah, and I would add that older males can develop relationships with females so that they're kind of around when she's ready to go for it. So it's a different kind of investment than the young male fighting and showing off his stuff. And I think that's actually part of the play there is the, helping her with the calf occasionally. It's not a bad idea.
The benefit of wisdom is pointed out here, right?
There you go.
Right. All right. So one more. This comes from Liam. Since there are 18 pregnancies, by the way, is that a record, Denise? 18?
Well, now we have two groups. So 10.
So it's hard to say. It's kind of apples and oranges.
Yeah, but it's a good amount for sure. So we got.
Yeah, I should say. So 18 pregnancies, this number higher than normal, which you just alluded to, does the hurricanes impact, do the hurricanes impact the number of pregnancies? Have you ever been able to connect the dots there?
Well, I think we looked at it, didn't we, Michelle? I think we looked at that and it wasn't what you would think. It was kind of the opposite. Yeah. Yeah. Well, remember that, if they're recovering from a hurricane, maybe there are changes in food or their group. So maybe it's not a good idea to get pregnant when you have these big environmental disruptions.
We saw a bit of a lull. We thought, after the hurricane, and there was quite a bit of loss of animals that you would just have a booming year the next year. And we saw a bit of a lull as they sort of recovered from it. So it took a little while and a few years until we got to, a big year of pregnant females.
All right, let's move on to our next WDP All-Star, who happened to be on the Stanella when I had the good fortune to take a voyage a couple of years ago. Leah McPherson, who in 2014 began as an intern and kept coming back like a lot of the people do with Denise and her band of dolphin chasers. And she's like all good ocean researchers. She's in Hawaii too. I'm really jealous. I totally made some bad career choices, clearly. So Leah's going to tell us about some new tools, which are kind of near and dear to my heart, how drones are coming into play as a tool for gathering data. Let's watch her video and then we'll bring her into the conversation.
Hi there, my name is Leah McPherson, and I'm tuning in from the island of Oahu, where I study Hawaiian spinner dolphins with the Marine Mammal Research Program at the University of Hawaii. I'm going to shift gears a little bit and talk about unoccupied aerial systems, commonly called drones, and how I'm using them both in the field with the Wild Dolphin Project and for my work here in Hawaii. I started flying drones in 2018 after being inspired by drone footage that a collaborator captured of the wild Atlantic spotted dolphins that we study. How could this bird's eye perspective change the way that we study dolphins? Let's look at some footage. So here you can see a group of spotted dolphins interacting with each other. You see a head-to-head behavior here, a little bit of pursuit, a little tail slap in the front. You can see individual movements and interactions between individuals. You can see how dolphins are responding to each other. This perspective also offers us an opportunity to study things like leadership and group formation. By following traveling groups of dolphins, we can see which individuals might be leading in a group or initiating direction changes. We can also observe if group formation changes with group composition, environmental variables such as water depth or habitat, or even in the presence of potential threats such as sharks or anthropogenic activity. You can see in this video a group of dolphins traveling over a shallow reef habitat, and they're fairly spread out, and the same individual tends to be in the front the entire time. In this next video, you can see the same group of dolphins, but the group is much more tight, and they're moving in deeper water. This is just one example of what we can see with this perspective. As we collect more and more of this data, we might be able to see trends in how group formation changes in response to certain things. This data becomes even more valuable because in many cases, we're able to identify individuals in the drone video. That's the beauty of studying dolphins with unique spot patterns. But drones are useful for studying dolphins and other marine mammals without spots too. One of the coolest techniques is called photogrammetry. So this is the drone that I used to capture the footage in the first two videos I showed you. It's called the Mavic Pro, made by DJI. And it's a great little drone to just throw up in the air and capture things like behavior. Easy to travel with too. One that I use a lot here in Hawaii that's not so easy to travel with is called the Inspire 2, and it's obviously much larger. The reason we use this drone is because we custom fitted it to have what's called a LiDAR altimeter. So this little unit here and this here on the bottom, can you see it? There we go. Actually send out a little pulsed laser that reflects off the surface of the earth, or in the ocean in our case. And that laser bounces back up to the unit and shows us how high we are in the air, so the altitude. And if we know the altitude, the focal length of the camera, the size of the image sensor, and the width of the image that results in the video, so the pixel length in that video clip, we can use it to measure the length of marine mammals, like spinner dolphins. Part of my project here on Oahu involves flying one of these drones over spinner dolphins as they surface to measure the lengths of dolphins in the population. I'll eventually be able to look at the proportion of small and large animals to estimate the age structure. This type of data can tell us a lot about the health of the population. In the next few years, I'm hoping to combine methodologies I've used with both the Wild Dolphin Project and the Marine Mammal Research Program to see if we can estimate the age of dolphins by measuring their length and body coloration. For example, in the first few years of life, Atlantic spotted dolphins grow in length and then they begin developing spots. Once they reach their full size, they continue to gain spots, in particular the white spots on the tops of their bodies. So while we can use length measurements to capture the age change for those younger animals, I'm hoping to use changes in coloration to continue capturing that growth when they're in their adulthood. I'm hoping to measure the percent coverage of white spotting on the backs of these dolphins to see if we can accurately estimate the age. We know the age of many individuals in the population of Atlantic spotted dolphins that the Wild Dolphin Project studies. Hopefully, it'll eventually allow us to estimate the age structures of other populations of spotted dolphins and marine mammals that change color as they age, such as Pantropolis spotted dolphins, Riss's dolphins, and even narwhals. So drones can be used for so many things in the field, and that's just a couple things I wanted to talk about today.
And Leah joins us live, and that's the view out your window at your place in Hawaii. Is that right?
Nope. Actually, this is Bimini. This is the Bahamas. I thought I would have a little bit of home.
Of course. It's just a little taste of home there. So, Leah, first of all, thank you, by the way, to people who've been donating. Jeannie Rice Camp, Rebecca Smith just chipped in a little bit. I got to ask you, as someone, as you know, I fly drones and frequently put them in the water, as you know, personally. But
one time,
one of the things, yeah, if that were only the case, one of the things about drones, as effective a tool as they are, is the drones you're using have limited duration, 30 minutes or so that you can fly them. And I imagine it would be really cool if you could get like something with a little more endurance. Have you looked at that? And this might be an opportunity for folks to support your research by helping you get, maybe a little bigger drone.
Yeah, definitely. The trade-off with that is that obviously these things, they're fighting winds, you're flying them, you're keeping this heavy thing in the air. So they use up a lot of battery power. The Inspire 2 takes 2 pretty large batteries. They're each about this big. And yeah, the flight time is a little under 20 minutes usually. So I think it's tricky. So the longer flight time you have, you're going to have larger and larger batteries, which is going to make it heavier and use more battery power. So I think everyone wants drones to be able to fly longer for, lots of purposes. Certainly that would be nice for doing focal follows and kind of tracking movements instead of having to land and swap batteries and relaunch again. So for behavioral things, especially, that would be very cool. But I think just the technology with these companies, it's not quite there yet, but we're always watching to see, if they come up with something new that has better battery power.
It's always about the batteries, isn't it? It's just with everything.
Yes.
Yep.
So drones clearly rock. They're an amazing tool. Marine mammal researchers have not regularly had aerial views of these animals to look at things like group structure. And one other cool thing, people are using them to look at the health of animals, the pregnancies, the width of an animal, that sort of thing. So Leah, what's on your wish list? What do you, besides batteries, what do you wish drones could do in the future that you would like to see?
Oh man, I mean, it would be cool if we could do biopsies from a drone for pretty elusive species. That's kind of, I don't know, obviously that would be very difficult. They use it for collecting breath samples, which is very cool. So when dolphins and whales exhale, and that kind of spout of, it's mostly seawater that's sitting on top, but also some snot in there. They can get a lot of interesting data from like bacteria and things that are in their lungs and data like that. But I think, I don't know. I mean, the most important thing on the drone is honestly just the camera. So I think better and better cameras. Also, you saw how large that drone is that I use here in Hawaii. If we could be able to have a good enough quality camera on a smaller drone, I think that would be really useful. Because right now, especially if you're following, an elusive species, there are some species out here in Hawaii that we don't see very often. And when you find them, it's like, oh, quick, get the drone up. But it takes a while. You have to put the propellers on. Whereas the Mavic Pro, like I said in the video, you just turn it on and throw it up in the air. So if we had kind of the LiDAR and camera capabilities on a smaller drone, I think that would allow us to just be more effective in the field.
Well, they get smaller and smaller. It's, interesting though, with cameras though, the chip size is so crucial that you're limited in some ways if you want to get the kind of resolution you need. And you sort of touched on this, but I am curious about this. If you could, do some fancy flying, the question from Rebecca is, could we get spout samples from the dolphins, or since they're smaller than whales, can we get, how close do you have to be? Just one other way to get data. And is that something that could you even imagine doing that with a drone? You could fly it through the spout. Would that be doable?
That people do that. Yes.
They do. Wow. That's really cool.
Personally, but for whales, yeah, it's called.
Don't let me do the flying when that happens. I think that'd be a bad idea, right?
Well, it would get wet.
It's called the snot bot. That's what they call it.
Yes. Yeah. So if you search snotbot, they've been doing it for a few years now, mostly with larger whales. But also
I got to do a story on this one. I love this.
It's very cool. Yeah. But they've done it with like beaked whales and I believe some blackfish too. And I think with dolphins, with smaller dolphins, like the Atlantic spotted dolphins that we have or spinner dolphins, they surface quickly. So it would be more difficult. And honestly, I think you could put like a little petri dish on a pole while they're bow riding and get it that way.
Yeah.
But with larger marine mammals, certainly they do that.
Good. Oh, and I want to shout out to Ruth Petzold, Evie Kimmel, Harvey Specter, all donating and ponying up for, what is it? Snot cam? Snot? What is it? Snot?
Snotbot.
Snotbot. All right, this has been kind of a racy discussion. We've been talking about shit and promiscuity and now snotbots. This is, it's kind of PG. So, all right. So, and Drew's out there just to razz me, asking if there are waterproof drones. Thank you, Drew, because Drew was there when I committed some aviation blunders there. But is there, can you get a drone that has either airbags or parachutes or some way to preserve it? I assume you've never flown one into the water.
I have not flown a drone into the water. I have had a drone had a firmware malfunction and fall into the water on me. Actually, the first day of my field season out here in January, which was horrifying as a new graduate student. However, I sent them the flight data and they said, "Oh, that wasn't your fault," and they sent me a new one for free. So I know it's not my fault. Good. I felt better about that. But to answer your question, they do have some water-resistant drones, waterproof drones. I think there's one that's called a splash drone, and I'm pretty sure it's meant to land on the water or get wet. So it's designed for that. And then I think that's the only one I can think of. And as far as parachutes go, they develop parachutes for larger drones. They're fairly pricey, but like the Inspire that I fly, I know they make a parachute for that one where it reaches a certain kind of spin and velocity, it'll deploy just automatically.
Yeah. I mean, I suppose you could find a long range, maybe a fixed wing drone that you could really travel with them on, maybe. But do you get enough data in the short spurts that it's okay, or would it be that much better if you could follow them for hours?
I mean, it really depends on what you're looking at. So for the photogrammetry stuff that I'm doing, it's not a huge issue if it's short flights because I'm really only measuring them when they're surfacing. So I can launch the drone, get them surfacing a couple times. If it's a larger whale, we can kind of get an idea of what the whale's downtime is. And if you're like, okay, this whale has been going down for 8 minutes at a time, then you time it when it goes down and launch the drone 6 minutes after it goes down. And then boom, you have the drone right up when it surfaces again. So since it's okay if it's punctuated for that kind of work.
All right, we're going to bring in everybody else to do some general Q&A here at the end of our program. And I'm just going to insert this question from Harvey for the Hawaii people. There have been some new rules implemented in Hawaii in regards to people swimming with the spinner dolphins. Can you elaborate on what the new rules are and what do they mean for researchers? In other words, if these rules apply to the Bahamas, would that be a problem for the Wild Dolphin Project?
So yes, very recently, this has been a long time coming, actually. People have been trying to protect spinner dolphins for over a decade now, and they have finally gone and made a federal rule for the 50-yard recommendation for swimming with Hawaiian spinner dolphins. And so previously, of course, the Marine Mammal Protection Act applies to all marine mammals in US waters, but it was a recommendation that people should not obviously chase and harass or hurt animals, but to approach them within 50 yards was not recommended. But now it is actually a federal law that you cannot approach and harass spinner dolphins, swim with them within 50 yards, or I guess it's going into effect at the end of this month. Which, yeah,
well, I think it'll be good for the
protection.
That sounds like a real problem. I hopefully the Bahamas isn't thinking of doing that, are they, Denise?
Well, the United States has had great marine mammal protection for decades, since 1972. The Bahamas do not have rules about getting in the water. We've tried to talk to them about various rules, and in the past we've had sort of educational discussions with other boats to try to minimize impact. And like in the United States, a researcher can get a permit usually to get in the water and do work. That's not such an issue because you're doing something besides just playing with, dolphins or whales. So that could still happen. Yeah, as far as I know, the Bahamas does not have that plan. that might be something down the road they want to implement in certain high traffic areas, because it could be a concern for sure. And I've been in different parts of the world where it's a nightmare with the public and boats. So you definitely want to protect animals before it gets too bad is the problem. Once you get industry started, that's when it's hard to regulate.
Yes, and that's an issue here as well. I mean, I think in the future, it could be a good thing for the Bahamas to consider, but the effects of like the human impact on animals here on spinner dolphins, just from what I've seen in both places, is much more. It's much worse. These animals don't get a break. On the Kona coast of the Big Islands, there are people around them over 83% of the time, and they're trying to rest too. So the spotted dolphins, they do rest during the day on the sandbanks, but they're also socializing. It's not kind of as strict of a resting pattern as the spinner dolphins in Hawaii have. So the spinner dolphins, they are nocturnal essentially. So they feed at night offshore on squid, again, kind of like the spotted dolphins. But then during the day, they go into this almost meditative, deep resting state. And, you know, they're easy to find. They're in these protected locations along the coastlines, and people swim with them. And, you know, my advisor likes to say people love them to death because obviously people want to experience these animals, and it's not a black and white issue. I'm all for people seeing and learning and loving these animals, but there's it has gotten to be a bit much in certain places.
It's a really tricky one, having had the thrilling experience of swimming in close proximity with dolphins as I have. And I recommend this to anybody out there. And certainly Denise and her team, these people take great respect and caution, and are not in any way harassing the dolphins, at least to my view of things. But as you say, if you have too many people doing it, but the flip side of that is that it really creates a tremendous amount of awareness and a desire to protect them. So I don't know how you settle that one. Denise, do you have thoughts on that one?
It's tricky.
Or go ahead if you want, Leah, go ahead.
No, go for it, Denise.
Well, you know, listen, it's 2021. We have a lot of different ways of educating people now. You know, we used to depend on marine parks. Now we have films, we have virtual interactive options. We have safe wildlife viewing. That's something that's enacted in the Keys, for example, where you get a seal of approval, right? That you follow the rules. So you can do something like that to minimize. Of course, you want people to educate, but I mean, we live in a culture of selfies that animals die because people are grabbing them and taking selfies. I mean, it's obsessive. So if you want to love and protect them, you might think about being hands off, you know, I mean, definitely.
It's a tough one. All right.
We have a big problem with that up in the Pacific Northwest with seal pups, and people want to take selfies with the seal pup, and then the seal pup gets left because the mom doesn't come back because you've touched the pup, and so she won't come back now. So
so you could even just touching a pup creates a scent that the mom will stay away from. Is that it? Or
if she could see you if she's out in the water, but there's something there that prevents her from oftentimes coming back, and then the seal pup basically dies unless a research group comes and brings them to a hospital or something like that. But it's those simple acts that people don't think are really impacting the animals that very much do.
Yeah. Well, here we are. The all-stars are all together here. I'm curious. I always like to ask what the dream project that you have in your mind right now, that if you had, if money were no object. So let me start with Cindy. If somebody watching right now ended up a big chunk of change, what would you do with it to advance this science?
Oh my gosh. That's a big question.
So many things. Yeah. Right.
I know. Right. Well, I mean, I think one thing is that our field in general is long-term research is not funded as well as some of these short-term projects. And so the idea to be able to have a long-term thing that's funded for many, many years, that you don't have to worry about every month or couple months trying to make sure you have enough money to keep going, to be able to do these long-term work and have this long-term data set, all the information and all the work that has been done with all these great colleagues and people is because Denise has been there for 30 years and we have that backlog data. I would not have been able to do my dissertation looking at what happened before and after the hurricanes if we didn't have those 20 years before. We wouldn't have known that there was a difference in the first place. So just the ability to keep going like that and have the ability to pay the researchers to be out there to get this information would be huge. We'd be able to do everything we wanted to if we could have that.
All right, Brittany, what's your dream piece of research? Oh, you're muted.
You're muted, Brittany.
You're muted. Yeah, unmute yourself, please. Go ahead. One more time. What's your dream?
My home range study was specific to the summertime when we're out there working on the boat, May to September. I'm really curious about that off season in the winter. And I know there are challenges with weather, of course. But if we had all the funding to, set up something in the Bahamas where we could go out and monitor the population in the winter, I think that would be really interesting.
I want to do a quick hello, by the way, to the folks watching on YouTube Live. We hope you're enjoying this program and you're learning some things. And ultimately, you're helping fund this really fascinating research. No one else in the world does this. All right, Michelle, what's your dream? Does it involve snot or fecal matter or what? What's the next bodily fluid that you want to go after?
I just, it seems like working with poop is like low key, sort of not all that expensive and intricate, but the work itself is very time consuming and expensive. Like genetic equipment is just very expensive. So for me, when I get held up with funds, it just means that I can't quite process all of the samples that we might want to process. Ultimately, what I would love to do is make sure that everything is processed, all of the individuals are genotyped and sampled from not just Little Bahama Bank, but all the new animals now that they're integrating with down in Grand Bahama Bank. And then along with that, this is a fantastic group of scientists, and I got to do this because I had support as a student. And so part of what I want to do is continue to get more students to learn how to do this type of work and get them excited about fecal genetics of all things.
You don't know how exciting it is when you're a graduate student. I know all you guys know this. The first time you get a poop sample, you come to the surface, you raise it up, and everybody on the boat is like, yeah, like it seems weird, but it's super exciting.
Wow. That's and just a quick thought here. I'm gonna come to you, Leah, with your dream research. But before I do that, you mentioned reaching out to other scientists. We have an all-woman panel here, and science is not known for being heavily populated with women. So, is this because Denise is awesome and you gravitated to her? Is it this field? Or is it that we're seeing a trend where more and more women are entering scientific fields? Denise, maybe you should take that.
I think to some degree,
Cindy, you want to do it?
Well, I think to some degree, some of it is the field that we are gravitate towards this more than men do. Most of my volunteers here in the Pacific Northwest are women as well. So I think there's some level of that.
Denise, any thoughts on that?
There are plenty of men in this field. I think our particular project, women are drawn to it, maybe because you are actually, close to the animals. But the fact that they can do science, which is great, and process data, it's sort of a win-win, right? I guess that's.
So if I may interject, we got the
Yes, please do.
Yeah, we had the opportunity, the fantastic opportunity to be mentored by one of the greats in the field. So Denise, she's an incredible person to work with and to learn from. And so she, for me, was a major draw into this field as well. It wasn't necessarily dolphins, but really finding a strong female scientist that could teach me some of the ropes.
There's Goodall and there's Fossey and there's Herzing. These are women who have dedicated their lives in a very specific way to understanding these populations in ways that no one else has. Okay, so by the way, thanks Ruth Petzold, Evie Kimmel, more donations coming in. We're at $6,750, folks. We're more than halfway there. We're going to go back now to Leah, and she's going to tell you how much her favorite drone is going to be for her next research project. And whatever it is, I want somebody to pony that up right now. No, Leah, what is your like dream piece of research that you'd like to do if you had the money?
Oh man, I mean, there's so many ideas, so many things to do in the future. I think it would be great, kind of, I touched on it a little bit at the end of my talk, to really do the photogrammetry with the spotted dolphins of like the whole population. So we can really ground truth the method and develop these new methods that we can then, then we take that as the case study and you take it to some critically endangered population of animals and you can just rapidly assess population health. I think that would be helpful for our data set here and super helpful generally, like to bring this out and help other populations that need the help. And then the second part of it, just because I'm here in Hawaii now with spinner dolphins, all of my work is boat-based and drone-based, but it's pretty cool to see, to like hear ear or sound trap recordings, and then even from the surface see what's happening, because behaviorally, I've noticed just a ton of similarities between spinner dolphins and spotted dolphins in the Bahamas. So I would love to do some type of like behavioral comparison between the two groups, just because they do have these similar patterns. I mean, I can look good down and be like, oh, that's aggression, or like that's water cavitation from a tail slap on the spectrogram. It's just like these things that the experience with the Wild Dolphin Project has really just like opened my eyes to, so that I feel like out on the water, even though I'm not doing behavioral science here, it's obvious there's so many similarities. So I would love to explore that.
Excellent. So now I have been a little bit neglectful on the silent auction issue. I apologize for that. You on the page there, click silent auction. You missed your chance to name an Atlantic spotted dolphin, but I have a hunch that if you really wanted to, we could make this happen. There's seahorse and shells, watercolor. There's another photograph which done by Island Exposure, which I believe just sold, but there could be more of those potentially. And then another photograph, Dinner Bell, from photographer Greg Dietrich. And then finally the oceanic white-tipped shark out of the deep blue. It's a stunning photograph mounted on metal, well worth the suggested bid there. So take a moment before you log off and maybe chip in a little bit more and maybe bid on one of these silent auction items, and all of it is going to a great cause. Denise, what's it like, even though it's virtual? It'd be nice to be in person, obviously, but it's nice to see all these faces, isn't it? Yeah, I assume you miss them a little bit, but I know you stay in close contact.
Yeah, but we miss them. They're not necessarily physically on the boat. And you didn't ask me what my ideal thing would be. With
yeah, of course. Geez, of course. Yes, it's kind of important.
I've always had ideas. And of course,
yes,
besides great big toys and cameras and sound equipment, I would like a bunch of money so I could clone this dream team.
I like that.
I could use a clone.
All around the world to study dolphins everywhere.
Well, the first thing we have to do is get their DNA from their fecal matter. So if you'll send that in, and then then we can begin the cloning process, right? Oh man, I took it dark, didn't I? Sorry about that. Denise, what's assuming we're completely out of COVID, let's hope, God forbid, we don't have another variant, but what are the plans for the next season?
Well, we always do our regular life history, reproductive monitoring every season. We're really excited. Next year, we were hoping to have it this year, but it didn't work out. Matthias Hoffman-Kunt, a colleague, we're going to have his underwater localization array finally to figure out who's talking to who, which is great. Much needed equipment, and looking forward to years of data collecting with that. That's on the board. And yeah, just other projects, maybe some sound traps for 24/7 recording. Perhaps that could be a great thing, and hopefully some fecal collection.
Excellent. By the way, let's show. We didn't show, as I was talking about the silent auction, some of the photos that are available. I think we're going to show one right now. Is that right? And I don't know which one it is.
Yeah, hey everybody, we're going to show you one of
here we go. Which one is that? Oh, that's
Dinner Bell.
God,
It's the dinner bell.
Dinner bells. But look,
nice.
is it you guys? That's Melissa and Cassie. Is that that's mounted on that kind of aluminum kind of, yeah. Oh God, it's really a spectacular printing process, and I highly recommend it. They're just gorgeous. They're works of art for sure, and that's a beauty.
They last forever. They're metal, so you can't really beat them up too much, and they don't fade or crack or corners chips or anything like that. And it's pretty large. You can see it's pretty big and it has a big bracket on the back and a way to hang it. And it's by a local photographer named Craig Dietrich. So he does some great wildlife photos. So.
He does a great job. And this is starting, I think, Melissa, you could probably get a gig at QVC. You're very good at this. This is good. So exactly. All right. So the silent auction ends in 40 minutes where we will keep that open. So go on there, peruse, and, get something cool, but more importantly, help out a really cool project. Denise, do you have any final thoughts?
I just want to thank everyone. You guys are awesome. As you know, probably don't say it often enough. And thanks for just working on the project and helping illuminate the mysteries of the dolphins. I mean, it's great. And you're all awesome young women with great minds and fun tools. And we're going to do more good things. So it's awesome. And that includes Cassie and Melissa, by the way, wherever you are back there. And thank you, Miles, as always. Great.
Always a pleasure. And Melissa Infante, Cassie Rucci, I should say, and Liam Groth behind the scenes. Thank you for all you did. Thank you all, those of you who donated. Really appreciate it. We didn't quite get to 10,000, but $6,700 plus is pretty good. And you can still donate anytime you like to keep the Stenella plying the waters of the Grand Bahama and Little Bahama Bank and doing this work that no one else in the world does. So it's a real thrill to talk to you all. I learned a lot, gave me a lot of ideas for potential stories. I think just it should be easy to sell the NewsHour on a story on dolphin feces, right? Don't you think? I mean, it's perfect. So
there's
anyway,
a whole soap opera you could do with everything that's going on.
Yeah, absolutely. It's a reality show. It really is. So, all right. Well, listen, thank you all very much. Thank you for being a part of this. And we're going to see you in the spring, maybe in person, maybe not. Who knows? We'll have to decide as we go. What do you think, Melissa? Maybe?
We're going to try. It's
okay,
4 months away till spring, so we're gonna see what we can
let's
do.
Hope. Yeah. All right, let's hope for the best, and so stay close to the Wild Dolphin Project. We'll keep you posted. We'll have some more to tell you about, and we really do appreciate your support. So thank you very much for being with us today.
Thank you, everyone.
We hope you've enjoyed learning about the Wild Dolphin Project and our work in the Bahamas for over 3 decades. And we hope you'll continue supporting our organization and other organizations that do critical work in the oceans and on our planet. Thank you.
If you would like to support us here at the Wild Dolphin Project, there are a few different ways in which you can. Donations are always needed and are a great way to help us out. You can also become a member, and with each membership level, there are different perks. You can also join us on a research trip where we have specific trips for interns and others for the general public. Another way you can help is just by spreading the word about who we are and what we do. And as always, please follow us on our social media sites and visit our website to keep up with what we are doing next.